Hello, welcome everyone. Today we have a very special guest, Professor of Molecular Physiology at the Leiden University, Mario van der Stelt. Welcome, Mario.
Thank you very much. Thanks for having me.
Great. Today we're going to talk about how endocannabinoids work in the brain and learn how to use the endocannabinoid system for medical applications. And before we start, I'd like to share some house rules with you. Questions can be asked via the Q&A. You can find the icon at the bottom of your screen. And at the end of the session, we will answer them. The webinar will be recorded and can be viewed on our YouTube channel later on. Let's begin with a poll to explore the knowledge level of our audience. As today's topic is quite in-depth, we would like to know your knowledge level on two topics, the endocannabinoid system and anandamide. Do you know what it is? Please answer your questions and the results will show in a minute. So what's your knowledge level of endocannabinoid system and anandamide? We will see the results hopefully in a minute. Here we go. So with regards to endocannabinoid system, the knowledge level is okay. I know something about it, 60%. I know a lot about it, 27%. So it's okay, but still we can discover more about it. We can learn more about it. And another one, anandamide, 53%, I know something about it. And 33% never heard about it. So still a lot to learn, I guess. Thanks for letting us know. Well, Mario, you are a professor of molecular physiology working at the Leiden Institute of Chemistry. And before we dive into the main topic, what does a professor of molecular physiology actually do?
Yeah, thank you very much for this kind introduction. And before I will tell you about what I do at Leiden University, I think perhaps it's nice to give a little bit about my background. So I studied chemistry at Utrecht University, and I did also my PhD. And already during my PhD, I was interested in the endocannabinoid system. Actually, I studied whether THC, so the psychoactive compound in marijuana, could protect the brain against acute brain damage. And we could show that that was indeed the case in animal studies. And after my PhD, I did a short postdoctoral research at the lab of Vincenzo Di Marzo. I guess some of you might have already heard of him. He is one of the world leaders in endocannabinoid research. And there I studied in depth what one of the endocannabinoids was doing, actually, not on the cannabinoid CB1 receptor, but on an ion channel called the TRPV1 channel. And this is the channel that's also responsible for the hot feeling after you eat a hot pepper. So anandamide is not only binding to the cannabinoid CB1 receptor, but also binds to an ion channel that gives this sensation of hot chili peppers. And after my postdoctoral stay, I worked for almost 8 years at a pharmaceutical company. I came back to the Netherlands and I became a group leader in medicinal chemistry at Organon in Os. And also there I studied the endocannabinoid system, and we actually developed some ligands for the cannabinoid CB2 receptor, for which we will discuss also that later. But yeah, maybe you've heard at that time, 2011 and 12, Organon was bought by an American company, and they decided to close the research in the Netherlands, the research site. So I decided to move back to the university. So in 2012, I started at Leiden University, and I became a medicinal chemist. And there I started to combine actually what I've learned at the company, at Organon, together with my previous experience of the research of the endocannabinoid system. So nowadays I've been working here for almost 10 years, and I've set up my own group, and that's the group of Molecular Physiology. And in my group, we have both medicinal chemists, so chemists, but also biologists. And together we try to develop chemical tools to answer biomedical questions. So we have also a lot of biologists in the group, and our main focus is indeed the endocannabinoid system. We are particularly interested in how the endocannabinoid system works in the brain, and also with the hope in the end to discover at some day a drug candidate that can be used to treat patients. And the other main topic in my group is kinase research for treating cancer. So these are the two main research lines in my group. So as a molecular physiologist, we try to understand the human biology and the brain. How does the brain function? And in order to answer these very difficult questions, we developed novel chemical tools, so small compounds to interfere and to visualize actually the proteins which are involved, for example, in neuro communication between nerve cells. And that's also why we are so very much interested in the endocannabinoid system, because this system is actually crucial to brain function.
Right. I can say you are a real expert on that. Before we dive into your research topics, maybe it's good to explain a little bit more about this endocannabinoid system. Yeah, how does it work?
Yeah, so I guess our audience is very familiar with medical marijuana. So marijuana is a product of the plant cannabis sativa, and that has been in use for more than thousands of years, actually, for its medicinal properties. And of course, the main popular effect is that you become high. So the psychoactive effect is very well known. And this is due because this cannabis sativa, this is an exceptional plant because it's the only plant actually that produces these type of compounds. So these compounds are called the cannabinoids, and THC is one of 80 to 100 different compounds that belong to this class of the cannabinoids. And THC itself is the only one that is responsible for these psychoactive effects. And that's because this THC is able to bind to a protein which is found in the brain, and this is called the cannabinoid CB1 receptor. There's actually another protein which is called the cannabinoid CB2 receptor, and this protein is found mainly in the cells of the immune system. So THC is able to bind to two different proteins, and actually upon binding of THC to these receptors, they're also called receptors, especially when the THC binds to the CB1 receptor, you will experience these psychoactive effects. So the CB1 receptor, which is highly expressed in the brain, actually this CB1 receptor belongs to the class of the G protein coupled receptors, a large class of proteins. More than 500 different proteins belong to this class. But the CB1 receptor is the most abundant protein of this class in the brain. Yeah. And it's also found in many different brain regions. So it's found in the brain region which is responsible for the memory formation, like the hippocampus, but it's also found in the cortex. For the cognitive effects, it's found in the basal ganglia, where you can, where it modulates the movement, or is responsible for motor control. And actually, because THC binds to all those CB1 receptors in all different brain regions, you can experience many different effects actually of smoking cannabis.
Yeah, we have actually an image of how this endocannabinoid system works. I wanted to show it to you.
Yeah.
This is actually an image of the two receptors that exist in the human body. That's what you mentioned, the CB1 receptor and the CB2 receptor. One is found mainly in the brain, and one is mainly found in the immune system.
Yeah. So here you see in the middle, you see the typical 7 transmembrane helices of these receptors. That's typical for this class. And as I said, the CB1 receptor is expressed in all different types of brain regions and thereby responsible for different physiological effects. And now it's very strange, actually, that humans would have a protein in the brain that responds to a plant compound. Why would that be? That's not because we like to smoke cannabis from the beginning. So there must be some reason why this protein is actually found in all these different brain regions. And that's because the brain is also able to produce THC-like compounds. So this you can call the brain's own cannabis. So they're also called the endogenous cannabinoids, or in short, endocannabinoids. So the brain is able to produce a lipophilic compound. THC is also very lipophilic. So it's producing a fatty acid type compound that can also activate the CB1 receptor. Maybe we can show the picture of THC, the structure. One further. Yes, so here on the left, you see the structure of THC, so the psychoactive compound. And then on the right, you see the structure of these compounds produced by the human brain itself. And there are two main compounds, two main endocannabinoids. They are called anandamide and 2-arachidonylglycerol. And actually, THC was discovered in 1964. Its structure was elucidated by Professor Meshulam from Hebrew University. And Professor Meshulam also was the one who identified for the first time the endocannabinoid anandamide, as you can see there. He actually invented the name anandamide. And ananda stands for bliss in Sanskrit. And so it's an amide, as you can see in the chemical structure. So Professor Meshulam was the first to identify THC and anandamide. That's a real tremendous achievement in my view. The really nice thing is actually that we have two endogenous compounds that can bind to the CB1 receptor, so anandamide and 2-AG, and so they also both bind to the CB1 receptor in the different parts of the brain, but we don't know actually what the physiological function is of anandamide and 2-AG. Why would the brain have actually two different compounds that can activate the same receptor? And that's actually one of the main questions of my research group. Can we actually study why anandamide, what's the physiological role of anandamide, and also what's the physiological role of 2-AG? Are they similar? Do they have different functions? And why would actually the brain invest in the production and degradation of these two types of endocannabinoids?
Yeah. Can I ask a question in between? You said that anandamide, you can compare it to, or it has the same effect as the phytocannabinoid THC. But does anandamide then also make you high?
That's a really good question. We think it does, but we have never tested it actually. It is not allowed to synthesize anandamide and then give it to a person. So it's like with drugs, medicines, they are strictly regulated. So you cannot synthesize anandamide and give it to a human. So that experiment has never been done, at least not in a legal setting. So we don't know whether you if you give anandamide to a human, whether you experience the same feeling of being high. What we have seen, actually, we did that experiment ourselves when we give these compounds, THC or anandamide, to mice, and you do see similar type of behavior. So in animals, we do see similar effects of anandamide and 2-AG, and that's comparable to THC, actually. Yep.
Yeah. So what actually fascinates you is, why does the brain produce these two endocannabinoids? You still haven't found that answer, haven't you?
It's an ongoing quest. Of course, we have already some clues what the different roles are for these different endocannabinoids. So for anandamide, we know it's involved in anxiety and pain responses. And 2-AG is also involved, for example, in appetite. So it stimulates the appetite. So different roles become clear. And actually, you have to give both compounds to elicit the similar effects as THC. So anandamide has when you stimulate anandamide in the brain, you get part of the effects of THC, and also with 2-HG, you get a part of the effects. But if you combine them, then you see the full-blown spectrum of physiological effects also observed with THC.
Yeah.
So it seems that different brain regions or different types of brain cells actually produce and use anandamide or 2-HG. So they are not always working at the same time at the same location in the brain. So, of course, yes, you wanted to ask?
Oh, yeah, well, we still, the question is, how does the endocannabinoid system works? Is still, there's still an image I would like to share with you, because we still have to explain it a little bit more.
Yes.
How does it, how does these receptors, neurotransmitters, how does that actually work? So maybe you can explain it via this image.
So the brain consists of many different types of cells, and one of the most important cell type in the brain is the neuron or the nerve cell. And these cells are excitable. You can really easily excite them. Then a small current is going through these cells, and then when those nerve cells communicate with each other, they release a so-called neurotransmitter. So that's a small molecule messenger that is released by one nerve cell, and then it travels through the synaptic cleft, and thereby it reaches the other side of this cleft, and there's another nerve cell waiting to receive these neurotransmitter signals. And you can see that here on the slide. There is a receptor, another receptor, type of receptor that can identify or bind to these neurotransmitters. And then this second nerve cell is also being activated. So nerve cells communicate with each other through these neurotransmitters. And now the effect of this CB1 receptor is actually to modulate this communication between the two nerve cells. So the CB1 receptor is mainly found, as we call it, at the presynaptic site. It's found on the sending nerve cell. So when then the receiving cell is being activated by the neurons, by these neurotransmitters, they start to produce these endocannabinoids. So the receiving cell starts to produce anandamide or 2-HG, and this is then released back into this synaptic cleft, as you can see, and then it travels through this space between those nerve cells back to the CB1 receptor. And upon binding of this endocannabinoid to the CB1 receptor, the release of these neurotransmitters is being halted. So it's a negative feedback system. So it was a central dogma in brain research that brain cells could communicate only in one way, the forward way, like with these neurotransmitters. But so the endocannabinoid system is the first physiological system in the brain in which it's shown that actually the receiving cells send a signal back to these presynaptic nerve cells, so to the receive of sending cells, to actually modulate this neurotransmitter release, and thereby, because it's found in many different cell types of brain regions, it modulates the neurotransmission in many different brain regions, and thereby modulates also the physiological effects in many different areas, and thereby you elicit all these different effects also observed with THC. So they are retrograde messengers, as it's called.
Well, now everybody knows how the endocannabinoid system works. Thank you for that. You have, besides that, you have also been researching a synthetic version of THC. That's actually your specialism as well. That's been like the common thread throughout your career. So a synthetic version, besides the phyto version, the plant version, the human version, now this synthetic version. Why has this been your main focus?
Yeah, so many patients use medical marijuana to alleviate their pain or to get better sleep, or to, MS patients to reduce their spasms. But one of the adverse effects, of course, of THC is that you become high, so the psychoactive effects. So what we really like to do is to have the beneficial medicinal effects of THC, but without inducing this psychotropic side effects. So we want to separate the beneficial effects from the adverse effects, and that's we can only do by making synthetic compounds. So we want to make compounds that resemble in function THC, but without inducing all these brain effects. And we can do that in part by making the compounds really selective for the cannabinoid CB2 receptor. Because we have not talked yet about CB2 receptor, but this CB2 receptor also is activated by THC, and thereby it reduces inflammation. So in certain brain diseases like multiple sclerosis, there's an inflammation in the brain. And we think, and other people in the field think, that if you activate actually selectively the CB2 receptor, you can reduce the inflammation in the brain. Because the immune cells, they will, if there is an inflammation in the brain, the immune cells in the periphery will travel through the blood-brain barrier and then go into the brain, and there they will attack the sites where there's some damage ongoing. So, for example, in multiple sclerosis, the protective lipid layer along the nerve cells is damaged in an autoimmune disease, and then when this process is not stopped in time, this will exacerbate the damage, and there will be an ongoing inflammation. And we think actually that if you activate the CB2 receptor, you can reduce this inflammatory process in the brain, and thereby spare neuronal damage. But of course, if our compound is not selective, but also activates the CB1 receptor again, then you get these psychotropic effects as well.
Right.
So we want to have a compound that only activates and binds to the CB2 receptor and not to the CB1 receptor. This is one of our research lines. So we investigate how we can make these type of compounds. Yes.
So the CB2 receptor, just to repeat it, it is to affect the immune system.
Yeah.
Exactly. But isn't it dangerous to use synthetic cannabinoids instead of plant version, or maybe try to create the endocannabinoids system, or endocannabinoids, I mean?
Yeah, synthetic compounds, of course, they have to be carefully tested. So you have to make sure that your compound is only binding to the CB2 receptor and not to other proteins. So there will be a lot of research, and many compounds have to be synthesized and tested on selectivity. And before you can go to clinical trials, you have to do all kinds of toxicology studies to make sure that your compound does not induce unexpected and unwanted side effects. So this is like for any drug discovery process, a regulated process. So also for targeting the cannabinoid system, you have to make sure that your compounds are safe before you go into humans.
Because synthetic cannabinoids can be toxic.
All compounds are toxic, depends on the dose. So also THC is also toxic. Yeah.
So the body responds to it anyway.
Sorry?
The body responds to it anyway as a toxic compound.
Yeah. So you have to adjust and make sure that you can dose as low as possible. The lower the dose, the less toxicity you will get. So you have to make your compounds really potent, make sure that they are absorbed by the body in the gut, for example, that they survive the liver, because the liver is an organ that is specialized in degrading compounds. It's actually why the effects of THC, when you eat a space cake, the effects are less intense and take longer, because it has to be taken up by the gut and will be degraded by the liver. So when you smoke THC, you surpass the liver and the gut. So you have a higher concentration of THC immediately in your blood that will travel through the brain. So that's actually the explanation why THC is probably most of the time smoked. Yes, and of course for a drug, smoking is not allowed. That's not really a fashionable way or a good way to administer a drug.
It's
So
unhealthy, exactly.
Yeah.
Yeah. You're currently working on 16 different lines of research. That's quite a lot. You must have a big research team.
Yes, so we have in my group we have two assistant professors, 16 PhD students, and all have their own project. They collaborate a lot, so we really form a team. But we have many different lines of research. Yes. So not only endocannabinoids, but that's an important part of the research in our group. And we also tried not only to develop selective CB2 ligands, but we also tried to design compounds that actually prevent the metabolism or the degradation of the endocannabinoids. So in this way, we can indirectly activate the CB1 receptor. That's also an ongoing research line in our lab at the moment, yes.
Right. Shall we highlight two of them? You already mentioned MS, multiple sclerosis study. The other one is a chemotherapy-induced pain. What do these two conditions have in common? I mean, is there similarity?
There is a similarity between multiple sclerosis and chemotherapy-induced pain. Of course, in MS patients, there's also a form of neuropathic pain. And in both conditions, there's also some kind of inflammation ongoing. In chemotherapy-induced pain, they're the extremities, so the fingers or the toes. There's the nerve cells that are responsible for the sensation there. They get damaged, and this will cause this pain feeling. And there also an inflammation response is ongoing. So there are some similarities where you can make use of the endocannabinoid system to interfere and to reduce the pain response, for example. So I've already talked about the project for the selective CB2 receptor agonist, but we have also a project specifically dedicated to inhibit or to block the enzyme monoacylglycerol lipase. And this enzyme or this protein is responsible for the degradation of 2-arachidonylglycerol, the other endocannabinoid. And we try to prevent this degradation of this compound by blocking this enzyme, the MOGL enzyme, and thereby we increase the concentration of the endogenous ligand 2AG, and thereby it can activate the CB1 or the CB2 receptor. Of course, if you block this enzyme in the brain, and there are currently also clinical trials ongoing with a pharmaceutical company, and they test this compound in all kinds of neurological disorders. And then you get also the psychotropic side effects. But we, in our group, we have designed and developed a compound that does not enter the brain. So it does not cross the blood-brain barrier. And we have now a really nice compound that blocks the degradation of 2AG, and thereby we can actually show that in an animal model, chemotherapy-induced pain is reduced with this compound. And we can also show that this is mediated through the cannabinoid CB2 receptor. So this is a very exciting project that's currently ongoing, and we collaborate with many different partners, not only in Leiden, but also at NIH in the United States, and Professor Andrea Homan. So this is really a multidisciplinary research, yes.
And the ultimate goal is to develop a new medicine.
Yes, in the end, we hope that we can contribute to the discovery of new drugs and new medicines inspired by the endocannabinoid system, yes.
And that's what this webinar was all about. We've already reached the end of this webinar. I'll have to check if there are any questions in our Q&A. You can still ask a question. There's one from Tina Kahu. Can cannabis help Parkinson's disease? Is there a study ongoing related to this condition?
I think there have been several clinical trials, very small scale clinical trials, to study indeed the effects of THC or other components. But the trials, they are small and not really well controlled, because that's a problem usually with cannabis-based trials. You don't have really good comparators on control groups. So it's difficult to conclude whether THC or cannabis has any beneficial effects at the moment. There's no real scientific evidence to support that claim at the moment, but people are still investigating it, yes.
Okay. Are there any other questions to ask Mario van der Stelt? You can do it right now. If
I see
not,
in the chat there are some questions, I see.
What's Mario's opinion? That's right, from Connor O'Brien on the entourage effect.
Yeah, so the entourage effect was coined by Professor Schulen, and he has observed that anandamide, one of the endocannabinoids, is not produced by itself alone. There are many different compounds, lipids, similar to anandamide, that are produced at the same time. And the idea proposed by Professor Schulen that these other compounds were there to protect the degradation of anandamide. So they are also being degraded, and because they are degraded, then anandamide can survive. So they prolong the action of anandamide. I think that's, to a certain extent, that can indeed help, but to prolong the actions of anandamide, that has been shown clearly in animal studies. But these other compounds like palmitoyl ethanolamide and oleoyl ethanolamide, they also have their own physiological function and receptors. So I don't think they are only there to protect anandamide from metabolism. They also are produced because they have their own role. So it's probably a combination.
Well, thank you very much, Mario. The webinar has come to an end. If you would like to learn more about, for instance, the endocannabinoid system, we do have a brochure. It's called A Primer to Medicinal Cannabis. You can download it from our website, and there's also an explanation about these CB1 and CB2 receptors. Thank you very much, Mario, and thanks everyone for joining us. This is the last webinar before the summer break. If you are interested in our previous webinars, you can go to our YouTube channel for an overview. Once again, I would like to thank you and have a very great day.
Thank you.